Izzy, the floor is yours.
Thank you, Juan, for your kind introduction. I'm really grateful to the AMMF for giving me, first of all, funding this project and for inviting me to present some of my work. So my name is Issy. I'm funded by the NIHR as a clinical lecturer, which essentially means I spend half my time as a clinical pathologist and the other half of my time doing cancer immunology research, which is what I did my PhD in. So I'm very interested in the role of interleukin 17, which is a cytokine, in the development of lymphatic vessels, also termed lymphangiogenesis in cholangiocarcinoma. And I've just listed here, apart from the AMF, a couple of other people who funded me. So I'm going to start by giving a bit of background on why we're interested in immune responses in cholangio. I'll give very quick snapshots of what we've done with the project and talk a bit about where we think this can go in the future. So I'm not going to go through the public health bits on cholangiocarcinoma because we had a lot of that yesterday. But essentially, we know that within cholangiocarcinoma tumors, so here is a gross image of a cholangiocarcinoma tumor. On the right, you have the H&E or hematoxylin and eosin slide of the same tumor. We know that within these tumors, we see lots of lymphocytes, also known as tumor-infiltrating lymphocytes. And one of the interesting things is a lot of the risk factors for cholangiocarcinoma are immune-related. So conditions like hepatitis B, hepatitis C, liver fluke infection, and even primary sclerosing cholangitis, which is an autoimmune disease, involve quite a heavy immune infiltrate into the tumor or around the tumor. Now, one of the questions that really interests me is whether this is cause or effect. Are the cells there because you have a lot of inflammation going on and you're just going to have immune cells migrating in that situation? Or do you have the cells there because they're actually contributing either to the onset of the disease or the progression? So one of the first things I did when I started my post was I took a number of cholangiocarcinoma tumors that we had. So quite luckily in Nottingham, we had matched tumors and non-tumor tissue obtained from the same patient tumors. And I carried out very basic immunohistochemistry staining, which is the bread and butter of pathology. And I was interested in looking at the number of lymphocytes of different subtypes that were within the tumors compared to the non-tumor tissue. So on the left panel here, I've delineated the tumor islets in these with these black lines, and we can see lots of brown dots within this figure, and those essentially represent CD3 positive T cells. Now, CD3 positive T cells, CD3 is a marker we use to identify all T cells. We can see quite nicely that you've got lots of T cells sitting around the bile ducts, and in the non-tumor specimen from the same patient, it's essentially quite clean. You don't see as many T cells. And when we quantify that, you can see you have significantly more T cells within the tumor stroma compared to the non-tumor stroma. We took that a step further and looked at a particular type of T cell subset known as helper T cells. And you see that you have more helper T cells in the tumor stroma versus non-tumor stroma, as well as macrophages. Now, we know macrophages are very important in the tumor process, but I'm going to restrict my talk to talking about T cells. So yesterday, we did a lot of the talks alluded to the potential uses of immunotherapy in cholangiocarcinoma, and also the fact that we all know that immune cells are present within these tumors. And here I've just pulled up a couple of recent papers talking about how the immune milieu or immune cell atlas of cholangiocarcinomas is increasingly being investigated because people are thinking, could we use this as a prognostic marker in this disease? So now I'm going to move very quickly into the cell type I'm interested in, which is the Th17 cell. And this is a very simplistic cartoon from this really nice review from Weaver et al. In 2006. Now, the eyes tend to roll when I start to talk about different T lineages, so just stay with me here. generally speaking, you have T cells will differentiate into different subtypes depending on the environment and the cytokines or chemical signals present within the environment. So again, generally speaking, we have four major subtypes: the Th1, Th2, Th17, and regulatory T cells. Now, effector T cells are the T cells that go into an area and do the work, either clear viruses, maybe clear tumors. Regulatory T cells are T cells that switch those immune responses off. So if your body feels that your immune cells are too active, they will send in regulatory T cells to switch to dampen down that response. Now, talking about the chemical signals that you need for generation of IL-17 cells, you need interleukin 6 and TGF-beta, plus or minus a few others. Now, I became interested in Th17 because other studies have shown that you have loads of TGF-beta and interleukin 6 floating around within cholangiocarcinomas. So I wondered, is it possible that if you have loads of these floating around, you're going to get generation of IL-17 cells? And quite serendipitously, I found there were two papers in the literature where they had looked at IL-17 positive cells and correlated those with disease phenotypes in cholangiocarcinoma. What they showed on this top left figure was in patients who had low numbers of IL-17, so that's the blue line, low numbers of IL-17 positive T cells, those patients had better disease-free survival and overall survival, meaning after surgery, they lived longer and their disease stayed away for longer periods. And that compared to patients who had high levels of IL-17 positive cells, those patients did worse. Again, they also looked at correlation of IL-17 positive cells with something called microvascular density. So I want us to focus on the yellow dots. And they looked at lymphatic microvascular density. And this is a histological measure of how many new lymphatic vessels you have within a tumor. And they found that patients who had more IL-17 positive cells had more new lymphatic vessels in their tumors. These patients ended up dying quicker, and they had more lymph node metastasis and generally more aggressive disease. So this was very observational. And that then led to this question in my mind of whether there was a mechanistic link between interleukin 17, lymphatic vessel development in cholangiocarcinoma. So I tried to approach this question using a three-pronged approach: in vitro analysis, bioinformatics, and digital pathology. And like I said, I'll just give very quick snapshots of what we found. So for the in vitro analysis, with collaboration with Mireia Sueca-Gomez, who's in Anna Grabowska's lab at Nottingham, we were able to generate tumor spheroids. So rather than growing the tumor cells flat on a dish, we grow them in a kind of three-dimensional way. And we label these cells with a green fluorescent dye so we can watch what they're doing as we keep them in culture. We grew them with T cells. So these are T cells that we've taken out from the blood of an individual. And the question we were asking was, can we get T cells if we grow them with tumor cells in this way? This is just an example of the flow cytometry plot. So our readout is looking at flow cytometry. We pull out these cells, we pop them through a flow cytometer, and through that we can look at the quantity of IL-17 producing T cells that we have. And this is, so short answer, yes. If I talk you through this, we first tried with two different cell lines, CCLP1, PRH knockdown and control, and compared them to the positive control. So these are T cells which have been treated with the chemical signals that we'll know will lead to IL-17 production. And we saw that when we grow these cells with these T cells with different CCA tumors, we did get increase in IL-17 production. So that was quite exciting. So then I repeated it with another CCA cell line, RBE, and with the AKN1, which are a non-cancer cell line. So it was nice to see that you don't get as much IL-17 with non-tumor cholangiocytes. But we were quite surprised to see that with another CCA cell line, had very little IL-17 production. And then the question was, what is different between these three cell lines? And we found that these two cell lines have higher expression of a protein called PRH, which is what Kevin Gaston and Sheila work on. And through collaborating with them and with some of their other interesting data they had, we've now got this funded by the BMA Foundation for Medical Research to look at what PRH is doing with IL-17 cells in CCA. So that's really exciting. So the next thing we did for the bioinformatics pipeline is that we went through with my bioinformatics colleagues, we went through the Cancer Genome Atlas and probed for genomic data from CCA tumors. We were able to pull out about 3,000 differentially expressed genes. And again, quite exciting, we found that lymphatic genes and IL-17 signaling pathway genes were among the genes that were differentially expressed in these tumors. So this work is still ongoing, but this is just a snapshot showing differentially expressed genes. So everything in blue is downregulated, everything in red is upregulated, and the grays are not significant. So we've looked at different IL-17 pathway genes and looked at different lymphatic genes, and what we can quite clearly see is that we have significant upregulation in some of those genes. Now, the fact that some are downregulated doesn't necessarily mean that they're important. It might just be that they're not the regulation doesn't happen at the transcription level. It might be post-transcriptionally regulated. And what we're doing now is we're analyzing RNA sequencing data on lymphatic vessels, which have been treated with different chemical signals. And we're going to try and do an overlay of the gene pathways that we get from those experiments with what's already available in the TCGA. So hopefully that should be exciting as well. And finally, I'm not sure how well this is projecting, but we've, with our collaborators at Birmingham, we've done some high multiplex digital pathology analysis. So this is a whole slide image of a cholangiocarcinoma tumor, and we have stained this with 8 colors. And this high multiplex allows us to look at multiple things at the same time in the same tissue. It's quite a challenge to get this optimized, as you can imagine, but we got there in the end. So I'm just going to give you an example of what we found and what we're currently looking at. So one of the questions we asked was, we know that there's IL-17 in the tumor. Which cells are producing the IL-17? Because the two major producers of interleukin-17 are the T cells and the macrophages. And those two act very differently within the tumor. So this blue stain is a nuclear stain. So every cell that is alive will stain with blue. CD3 stains T cells, which is in green. IL-17 stains the chemical signal, so interleukin-17, that's in orange. And the macrophages are stained with CD68, which is in red. And when we merge the images, we can see quite nicely that the yellow stain, if we see this blue cell, which is the nucleus with the yellow around it, this is a merge of green and orange. So we have CD3 positive T cells producing IL-17, but not so much the macrophages, which are over here. And this is what we see in most of the tumor cells we've seen. So it seems that within CCA, the majority of the interleukin-17 producing cells are T cells, which is quite nice when your hypothesis seems to work out. The other question we're asking is, can we do neighborhood analysis to decipher whether you have more lymphatic vessel generation next to the areas where you have IL-17 producing T cells? Again, we have the nuclear stain in blue, the CD3 positive cells in green, IL-17 in orange, and podoplanin, which is one of the markers to identify lymphatic vessels that we use. And when you merge, again in the same area, you can see the IL-17 producing cells, but you can also see the lymphatic vessels sitting right next to the IL-17 producing cells. And this is quite interesting, and one of the things I'm really keen to do is establish collaborations with computational biologists and mathematicians, because what we want to do is generate really robust algorithms to make sure that the data we're seeing is real data. Because if you do multiplex imaging, you can analyze it in 1,000 different ways. And we want to be sure that what we're seeing is reproducible. So we have a test set of 10 cholangiocarcinoma tumors. And once we have optimized our algorithm, then the idea is to take that into larger numbers with our collaborator, Dr. Luke Bolter, who's based at Edinburgh. So just to quickly conclude, we've shown that T-cell polarization to an IL-17 secreting phenotype actually occurs in the presence of CCA tumors, and we've shown that in vitro. Our bioinformatics analysis has shown that the IL-17 signaling pathway and lymphatic genes are actually differentially expressed within CCA tumors. And finally, that IL-17 producing T cells are present within human CCA tumors and appear to co-localize with lymphatic vessels. Now, why do we care? Why is this important? IL-17 has been shown to be important in autoimmune diseases like psoriasis and rheumatoid arthritis. And anti-IL-17 drugs are already standard of care in quite a few cases on the NHS. So the idea behind this is if we can prove mechanistically that patients who have increased IL-17 within their tumors will do worse, and we can show why they do worse, then potentially we can have enough data to call for clinical trials, potentially of clinical trials of testing already existing anti-IL-17 drugs in CCA tumors. And I just want to acknowledge all my collaborators within Nottingham Birmingham and the people who funded this research. Thank you.